As the science of aging advances, one of the most exciting and transformative discoveries has emerged from the field of epigenetics, revealing that our genes are not destiny and that the way we live can shape how we age at the molecular level. Unlike mutations in DNA, which are fixed, epigenetic modifications are chemical changes to the genome that can turn genes on or off without altering the underlying genetic code. These modifications, such as DNA methylation or histone acetylation, act like switches and dimmers, regulating the activity of genes in response to our environment, diet, stress, and behavior. This means that every decision we make, from how much we move to what we eat, how well we sleep, and even our social connections, can influence how our bodies function and how gracefully we age. What makes this discovery so profound is that it helps explain why some individuals maintain robust physical and cognitive health into their 90s and beyond, while others experience chronic diseases decades earlier. The growing body of epigenetic research is beginning to show that longevity is not just about having the right genes, but about how those genes are expressed or silenced over a lifetime. In particular, studies of centenarians living in regions like Salento in southern Italy are shedding light on this relationship between environment, behavior, and gene regulation. These long-lived individuals often show unique epigenetic signatures that distinguish them from both younger people and from peers in other regions with lower life expectancy. Salento's centenarians, much like those in other longevity hotspots, have spent their lives immersed in routines that support biological resilience, eating whole, seasonal foods rich in plant polyphenol, walking regularly through hilly terrain, sleeping well, engaging in strong social networks, and maintaining a clear sense of purpose. These lifestyle factors appear to leave an imprint on their genome, not by altering DNA, but by shaping the epigenome. For example, researchers have found that these individuals tend to have more stable patterns of DNA methylation, particularly at genes associated with inflammation, metabolic control, and cellular repair. In aging populations outside these regions, methylation patterns often become erratic, leading to overexpression of harmful genes and underexpression of protective ones. In contrast, the centenarians of Salento show a kind of epigenetic balance that mirrors their overall physiological stability. This is not just a fascinating academic detail, it has real implications for public health and personal decision-making. Epigenetic changes are dynamic, and they can occur over relatively short periods. A person's biological age, as measured by their epigenetic clock, can be accelerated or decelerated depending on factors like diet, exercise, stress levels, toxin exposure, and sleep quality. Some studies even suggest that lifestyle changes can partially reverse markers of biological aging, slowing the ticking of this internal clock. Among the most compelling findings is the connection between inflammation and epigenetic drift, the gradual loss of proper gene regulation as we age. Chronic low-grade inflammation, often driven by poor diet, sedentary living, and psychological stress, can disrupt methylation patterns and accelerate biological aging. Yet this drift appears to be less pronounced in individuals who follow anti-inflammatory diets like the Mediterranean pattern and who live in tightly knit communities that buffer against loneliness and chronic stress. In other words, the lifestyle of long-lived Italians is not just protective in theory, it is modifying their gene expression in real time. Physical activity, another cornerstone of healthy aging, is also known to influence epigenetic mechanisms. Regular movement helps regulate pathways involved in energy metabolism, immune response, and tissue repair. It may even help maintain the integrity of telomeres, the protective caps on chromosomes that shorten with age, by influencing genes involved in cellular stress responses. Likewise, the natural rhythm of day and night, when supported by consistent sleep and exposure to sunlight, plays a key role in maintaining circadian regulation, which in turn keeps epigenetic patterns aligned with the body's needs. Perhaps most remarkably, these insights suggest that our epigenome is not just a passive reflection of our environment, but a potential target for intentional change. Interventions like meditation, time-restricted eating, polyphenol-rich diets, and even structured social engagement are being studied for their capacity to influence methylation patterns and gene expression in ways that support longevity. It is not far-fetched to imagine a future where epigenetic profiling helps guide personalized wellness strategies, offering each person a unique blueprint for healthy aging based on their molecular history and current habits. In the end, what the science of epigenetics tells us is both empowering and humbling. We are not merely the sum of our inherited genes. We are the stewards of how those genes are used. The daily decisions we make, what we eat, how we move, whether we foster connection or allow ourselves to disconnect, are inscribed into our biology, shaping the course of our health over time. The centenarians of Salento are not accidental anomalies. They are living proof that the aging process is not rigid but responsive, and that with care, attention, and intention, we too can influence the script our bodies follow as we grow older.